So in this next recording, what I want to do is go over 8 of the 9 phyla that we're going to talk about as representative of the animal kingdom. So we're going to go over Porifera, Cnideria, Platyhelminthes, Nematoda, Molluska, Annelida, Arthropoda, and Echinodermata, which is a lot. So this is going to be a little bit of a longer recording, but I want to use these words as much as I can so that you become familiar with them. And the only way to do that is you've got to use them. So talk about this stuff with your classmates, talk about it with other people, but these weird words like Platyhelminthes have to roll off your tongue, and that only happens when you use them. So let's start with the simplest of animals, and that is Porifera, phylum Porifera, and these are the sponges. So you're responsible for knowing each of these and examples of each. So Porifera are the sponges. They don't have a true vascular system. They're really only 2 cell layers with a kind of a jelly system in between. Kind of a very interesting animal. Originally, actually, sponges were thought to be plants, but in fact, they are animals. They meet all our criteria. But the basic body plan is a large tube with an opening at one end and is perforated by many tiny holes. So the actual sponges that we use now, most of the time, are artificial sponges. It's just chemicals. But originally, to get a real sponge, people used to just harvest these, and then the protein that makes up the structure of the sponge is called spongin, would be what you have that works as a sponge. Okay. Now, they're filter feeders, so they create a flow of water through all these little tiny holes with these flagella, these special cells with their flagella, and they capture tiny bits of organic matter and bacteria and consume them. So there are probably about 5,000 species of Porifera. Most of them are marine, so that means they are in a saltwater environment. So that's Porifera. Now, Cnideria is our next phylum, and the Cnidariids are the hydras and the jellyfish and the sea anemones and the coral. Now, the unifying theme of Cnideria is a stinging cell that they have, and that's called a cnidocyte. And we'll talk about that in just a second. But this is a hydra. You find it in fresh water. And on those long tentacles are tiny stinging cells that allow it to capture prey and then allow it to paralyze those prey so it can be consumed. Most of these animals are very delicate in nature, so they have to paralyze their prey so they can be digested without being torn apart. Think of a jellyfish, right? Which really aren't fish. We call them sea jellies, to be politically correct. But the basic form of the sea jellies, as an example, are a medusa form. Or you may see a sea anemone in the polyp form. And what both of these have is what's called a gastrovascular cavity. Now, the gastrovascular cavity is just that, gastrovascular. Gastro meaning kind of like our digestive system, versus vascular, which is our kind of system for moving fluids around. So these are like vases. You bring food in through a common mouth anus, so be thankful for the little things that you don't have a common mouth anus, like a jellyfish or a sea anemone. And then food is then digested in the gastrovascular cavity, and then diffusion is the main means by which that food and nutrients then distribute to the rest of the organism. Then anything that's undigestible then is expelled. They have a basic neural net, so we're starting to see a division of labor and the development of basic nervous system, and they are all carnivores. So this is now the idea that we have to put stinging cells on those very, very delicate tentacles because we eat other animals. So this is a picture of that cnidocyte, that cell that contains that very, very effective thread for the discharge of venom. So the nematocyst is the actual organelle. This is a very complex cell then, which is then triggered by touch. So the thread is discharged, it wraps up the prey, and injects venom. Right? So if you've ever been stung by a jellyfish or a Portuguese man of war, you know that it can be and is incredibly painful. So this is actually a picture from a website for a tourist place in Hawaii. And so they're like, beware when you swim in the ocean, depending on time of year, for Portuguese man of war and the box jellyfish. So some of these versions, the species of the box jelly actually that's found around Australia, can kill you from its venom. Sea anemones are in this group, as well as the coral. So those are the Cnideria. Now our next group are the Platyhelminthes. And Platyhelminthes are the flatworms. And we're going to look at three groups of flatworms. Free-living, the flukes, and the tapeworms. Now, the flatworms are named as such because they're all leaf or ribbon-like animals. Okay, there's a fluke in someone's hand. Now, the anatomy of a planaria is displayed here, and this is a great example of the flatworm. It's one of the most common ones that you'll see when you look at flatworms in the lab. Now, you have what's this gastrovascular cavity again, but you can see now it is much more extensive, trying to distribute itself through a much denser organism with a lot more cellular tissue here. You have a mouth or feeding tube, this pharynx, and you have cephalization. So we've gone from what was basically radial symmetry among the Cnidaria into bilateral symmetry with the flatworms, with the Platyhelminthes. So we have a very basic clustering of nervous tissue in the ganglia at the head and these eye spots. Now, these eye spots don't really see vision, but they can tell the difference between light. And so most planaria that you find in fresh water will swim towards the darkness, because if I'm in the dark, if I'm in the shade, I'm less likely to get eaten by something because it can't see me. Planaria also, speaking to their very simple form, have incredible powers of regeneration. So this is experiments that I don't do in lab, but people have done in lab, and I did as a high school student, where you can cut a planaria in two, and two new planaria will grow from that. You can actually bisect the head or the tail, and you'll get a two-headed planarian. Any piece of the organism, if it's of sufficient mass, will generate a whole new organism. Some advantage to being simple. Now, the planarian is our example of a free-living flatworm. Primitive nervous system, mouth and feeding tube. It has a phototaxis, negative phototaxis. It moves away from the light. So we're starting to move and sound more like an animal here. Now, the flukes are another group of Platyhelminthes, and these are parasitic. The example here is the blood fluke, schistosoma, and it causes a disease called schistosomiasis. So schistosoma is not something that you want to enter into a relationship with. And what it does is it actually attaches to the blood vessels near the intestine, and it lives off your blood then. It literally goes through a couple different organisms in its life cycle, snails being a different host. The larvae that are produced from the snails then actually penetrate the skin of humans and will then swim through your blood vessels till they latch on near your intestines. Unfortunate. Again, if you're interested in parasites, Parasite Rex is a great book to read more about them. Continuing in our vein of parasites are the third group of Platyhelminthes, the flatworms, and that is the tapeworms, which we saw the tapeworms earlier when we talked about a parasitic relationship. Tapeworms do inhabit our intestines, and they absorb our partially digested food directly into their bodies. On the anterior end, you see a bunch of hooks and suckers that allow them to attach to the interior of the intestinal wall and stay there. There have been times in history where people will purposely swallow tapeworms as a method of weight control. It is not recommended. We see the larvae of tapeworms found in raw meat, and this is one of the reasons why cooking our meat is an advantage in many cases, because it helps reduce the incidence of parasites that we introduce to our body, because it kills the parasite before we ingest it. Now, our next group are the nematodes. Now, the nematodes we've gone from the flatworms to the roundworms. And this is a picture of a very, very important roundworm. These are very tiny, millimeters in length, called C. elegans. And it is an incredibly important model organism in molecular biology because it has only 1,000 cells. So you will hear about C. elegans as a model organism, and you know that it's a nematode. Now, nematodes are found almost everywhere. So they are our third group of decomposers. So there are many, many thousands of nematodes you'd found in any typical healthy garden soil. So they're feeding on rotting organic material, and some are going to be parasitic and feed on the tissues of plants or animals. They have a complete digestive tract, so that's something to be thankful for. And so this we see now a true division of labor in the roundworms. Another example of a roundworm is trichinella. And trichinella is a disease, trichinosis, you can get from eating raw pork. So once again, an undercooked meat, this is a organism that you can ingest. Now, the other thing that's interesting about nematodes is this is going to be the first time that we see the evidence for a body cavity. And that's one of the trends that we're going to follow as we move through the different phyla. Body cavity is a space inside the organism that separates then internal organs from the body wall. Now, something that is acoelomate without a coelom would be like the flatworms, where the tissue is solid from the exterior of the organism all the way to the digestive tract. There's nothing, there's no space in there. Now, if you go into a roundworm, the nematodes, you're going to get a space. You can cut this thing open, like doing surgery on it. You can open it up, and then you can see the organs inside because there's what we call a pseudocoelom. Now, as we move forward into the Molluska and Annelida, we see what's called a true coelom, a coelomate organism. And this is going to have now internal organs suspended in the middle tissue layer. And so that's what we have. So when you undergo surgery, like in your abdomen, they're going to open up your abdominal wall, and then there's going to be a sac in which all of your organs are suspended. You have to cut through that then to get to your organs to help you if you need surgery. But that is a true coelom. What are the advantages of having a coelom? We believe that it adds significantly to the flexibility of the organism, plus it protects those internal organs. As an animal, we're not like a plant. We do move around. We run into stuff, we fight. Right? Sometimes we have to kill our prey and struggle with it. So the more protected we are, or protect those vital organs, the better off we're going to be. And so we think that that's one of the advantages of having this body cavity, this coelom, as we move into more advanced or more complex versions of animals. All right, so the next group that we're looking at is Molluska. And the mollusks then are going to break down into three groups: the gastropods, the bivalves, and the cephalopods. But a general view of mollusks we see here are much more advanced. We've got organ systems, we've got a brain, we've got gonads. The thing about mollusks is they all share a couple of similarities. Those being a foot, which is a muscular extension of the body, so think of snails or a clam. A mantle, which those that have a shell will be secreted from the mantle. This is a draping outgrowth, and that secretes a shell. So think of a snail or a clam again in that respect. And then finally, a radula, which is a rasping or file-like tongue. And so this is one of the reasons why you put snails into your fish tank, because they literally go along, creeping along, scraping the algae that's growing on the sides of your fish tank and eating it, and they use their radula to accomplish that. We find examples of mollusca in marine environments and freshwater environments, and also living on land. So think about scallop as an example of a bivalve, right? You can look at the cephalopods. These are the squid, a nautilus, the octopi, and then finally the gastropods would be snails and slugs. Right? So those are our three groups of mollusca, and that moves us on then to our next group, which is the annelids. Annelida. And Annelida now are our segmented worms. So the segmented worms we think of like an earthworm, right? So our earthworm then is much more complex. We see cephalization, we see a true coelom, and here now we have an organism that has somewhat of a repeat in its body segments. And so we're going to see that in the earthworm clearly, but we also see that in more advanced organisms that we'll see, and I'll give you examples when we get to chordates as well. So Annelida have examples that live in freshwater, marine, and moist earth environments. Earthworms are very important agriculturally. They literally eat their way through the soil, producing aeration in the soil, and that increases the health of the soil. More things can live there, and crop productivity. All right. We're going to see segmentation in arthropods and chordates as well. So this is another kind of step in evolution as we increase body plan complexity. Now, we're almost done. Our next group after the annelids, so the leeches are involved in the annelids as well. You see a marine polychaete, feather duster, all kinds of examples. If any one of these things are interesting to you, read more deeply. We're going to go over the groups because we need to memorize them in class and in lab, but this is our introduction to each of these groups. Now, the arthropods are our largest, most diverse group of all animals. The arthropods, there's nearly 1 million different kinds, and among the arthropods are the insects, and even the insects in themselves are more diverse than all the other groups combined. We're going to look at 3 different groups of arthropods: the arachnids, the crustaceans, and the insects. Now, all of these are known for their jointed appendages, right? So Arthropoda means literally jointed foot. And all arthropods then also have an exoskeleton, a hard external frame that gives them support and protection, and it's made out of chitin. So chitin, if you recall, is the same thing that fungal cell walls are made out of. Right? So when you have an exoskeleton, you have the muscles basically are attaching to the inside of the exoskeleton. They have an open circulatory system, and they'll also have to go through a molting process. When they start to continue to grow and their exoskeleton becomes too small, they'll literally crack out of it, be soft and vulnerable, and then secrete a new exoskeleton that is larger for them to continue to grow on. Now, considering our diversity, again, we have arachnids, which are scorpions, spiders, ticks, and mites. We have the crustaceans, which are our lobsters, crayfish, barnacles. And then finally, we have the insects. And this is by far the largest groups. So insects are known because they have our 3 body parts: a head, a thorax, and an abdomen. But more importantly, that they're so incredibly diverse. There's so many insects out there. You can look at the different orders of insects. Remember, dumb King Philip can only. So this is now below class. This is class Insecta. Orders Coloptera, Dermoptera, Hymenoptera. So lots and lots of different stuff. If you look, 500,000 different species of beetles, right? 120,000 different species of flies. Okay, 100,000 different species of ants and bees and wasps. Incredibly large, incredibly diverse. And one of the reasons that we think there are so many different kinds of insects is because of their coevolution with the angiosperms. So just using beetles as an example, some of the research that people have done have shown that when these groups started to feed on angiosperms, we saw a radiation in that species. That is diversification of the number of species in that group. And so you can see the green numbers here are beetles that feed on angiosperms as compared to beetles that don't feed on angiosperms, and the point at which they switched to feeding. So remember, coevolution is you have the evolution of 2 species that are interdependent, affecting the evolution of the other, exerting selective pressure on each other. Now, the other thing that often comes along with insects, but not exclusive to insects, is the idea of metamorphosis. So here now you have a caterpillar on the left, which is the larval stage or immature stage of the insect, and a butterfly on the right, a monarch butterfly. And so were you to go out in the field and say, oh, here's a butterfly, that's a nice organism, and here's a caterpillar, that's another nice organism, you may very easily say, well, these are 2 different species. But in fact, they're the same species. So when an organism goes through a significant morphological change, moving from a larval stage, an immature adult, to an adult stage, we call that a metamorphosis. And we see that very commonly in the insect kingdom. Now, our final group that we're going to talk about today, and then we'll spend the rest of our time looking at the animal kingdom, looking at chordata, is Echinodermata. So Echinodermata are the sea stars and sea urchins. And again, because they're deuterostomes, they're more closely related to us than we are to the other phyla, but they don't look anything like us. So again, you have to think about what are the factors that you look at to look at evolutionary relatedness. And so we look at that developmental system there. Now, this is kind of a weird group. It looks like they have radial symmetry, but it's not exactly radial symmetry, because the 5-armed sea star, you can't divide down any diameter to get 2 identical halves. It doesn't quite work that way. They do have a hard spiny surface, though it's not a true exoskeleton. They do have an endoskeletal-based body. They do have a unique water vascular system that allows them to hold onto rocks, their substrate, very, very, very strongly, and that allows them to live in these intertidal zones. So this water vascular system is many, many feet on the bottom, as you can see underneath these tube feet under each arm, allows them to grasp. Now, the main food of sea stars in many cases are bivalves, or clams, and they have a very unique way of digesting clams in that they force the clam open just a little bit, and then they invert their stomach out through their mouth and into the clam to digest the clam's contents, and then to swallow their stomach back in to complete the digestion and distribute the nutrients. Once again, the animal kingdom is very unusual. So just to review, these are the 9 phyla that you need to memorize. We've talked about now Porifera, the sponges, Cnideria, Platyhelminthes, the flatworms, Nematodes, the roundworms, Molluska, Annelida, the segmented worms, Arthropoda, including the insects, the most diverse, largest in diversity of all the phyla, and Echinodermata. The final group that we'll look at is Chordata, and that is how we'll wrap up the animal kingdom.